This is just showing that plants are surrounded by all kinds of organisms in their environment, so they coexist and live with different organisms that are in the soil or on their body or in their body, in their tissues. And so we've been looking at this a little bit. This one is actually looking at um, they're focusing in on the genes. And we'll come back to this idea too with how these other organisms, especially if they're intimately coexisting with the plant, that their genes need to also be considered when you think about what's going on with the plant and how do they survive in their habitat. And so we'll come back to this idea. But first, let's finish going through some of the organisms. I believe we were working with mycorrhizal fungi when we left off last week. And so we looked at how mycorrhizal fungi have this hyphal growth form, these long filamentous hyphal filaments that will grow on the root. Some grow into the root tissue. And then you can see they're also showing I guess they have it color coded here, the yellow and the orange looking stuff there. Those are the hyphal filaments that are going off into the soil. And so this picture, I think we looked at, and how the fungal hyphae can be it is much more petite than even the finest plant root. And that really can help the plant get access to areas of the soil that it itself cannot do with its thicker plant roots. I can't remember if we looked at this slide yet. So with mycorrhizal fungi, if you see the word mycorrhizae, like on the top there, that literally means fungus root. And so myco is referring to the fungus. Anytime you see RRHIZA, something like that, in a word, it's referring to something that is a root or something that's root-like. So this is a fungus root symbiosis if they use the term mycorrhizae. And then we talk about the mycorrhizal fungi that are able to form these partnerships. It's a win-win for the most part because the fungus certainly does get sugars from the plant. In fact, it can be quite substantial of what the plant will share. So if the plant is sharing a significant amount of its sugars, that really does point to the idea that this is a good situation for the plant. It's worth the trouble to feed the fungus. And the plant really does benefit. There's thousands of studies out there published showing that the plant is benefited by increased access to water in the soil. And I ranked these elements, phosphorus, nitrogen, and potassium. That's on purpose instead of saying NPK like most of the fertilizers you see, bags of fertilizer. I'm putting the P first because that's phosphorus. And so with the help of the fungus, the plant can get access to phosphorus that can be really challenging for the plant to get access to all by itself. So in the soil system, phosphorus is often complexed with other chemicals and it precipitates out. So even if you get your soil tested, the testing agency will go, oh yeah, there's phosphorus in your soil. But the next question is, but what's the available phosphorus for the plant? And that's a big distinction. And so the fungus is really good at getting access to phosphorus the plant cannot access. I'll talk a little bit more about that idea in a minute. And it also helps with nitrogen and potassium uptake, which the plants need all three of those in fairly high quantities. It overall, again, thousands of studies show that if you have plants without mycorrhizal fungi and the same exact plant inoculated, you can see the seedling growth is so different. It really stimulates the seedling growth and then all the way into maturity. And some mycorrhizal fungi are pretty good at protecting the plant from pathogens and actually actively helping to either inhibit or kill the plant pathogen. So from that regard, that's a definite bonus. And then many, many, many studies have been done that show the ability of the plant to survive if they have the mycorrhizal partner. They can survive salty soil, droughty soil, heavy metal contamination, and the list goes on. And so those are more like your abiotic stresses. Abiotic meaning non-living, something, the habitat is causing some situation. So the abiotic stresses, the mycorrhizal fungus can really help the plant. So going back to that first slide set, when we were looking at the process of how the plant went from the ocean environment onto the terrestrial surface, this idea that the green algae ancestor they've actually shown that green algae, some scientists say it was really the charophytes that the terrestrial plants came from. I'm fine with green algae right now because this is still hotly debated. So let's just say green algae were the ancestor to terrestrial plants. And then this paper that I'm showing the title of was published not too long ago, where they show that the green algae actually has genes necessary to form a relationship with mycorrhizal fungi, even though the algae is in the ocean and is not forming that relationship. So the paper, I'm not going to ask you to read the paper, but basically it's showing how they talk in this paper how the first terrestrial plant is really primed and ready to form this interaction with mycorrhizal fungi because part of its DNA has the necessary genes to form that relationship. And so I mean, if you think about this, this is such a major shift in the life and habitat of plants, to go from ocean to terrestrial environment about 500 million years ago. And without the fungi there, this would have not happened or taken a lot longer to happen. And so again, fungi are incredibly important, and it's really the mycorrhizal fungi that they point to. It's the mycorrhizal fungi that allowed for plants to actually make it in the terrestrial environment. Depending on who you talk to, they'll mention two different groups of fungi, Mucoromycotina or Glomeromycotina. You don't really need to know that for test material, but just know that they're actively investigating what group was the first. I'm fine with you understanding that mycorrhizal fungi, just in general, you don't have to know the actual group. But mycorrhizal fungi were necessary for the plants to make it on the terrestrial habitats. So if you think about all the different plants out there, there's a lot out there, and most of them are forming this relationship with these fungal partners. Depending on, again, who you talk to, some sources say 85% of plants. I've seen that push to 90. So 85 to 90% of plants that are on the face of the earth form this relationship with mycorrhizal fungi. And about 72% are what we call the arbuscular mycorrhizal fungi. And you can see with the pie chart, that's the deeper blue chunk of it. So the vast majority of plants have that relationship. 10% orchids have their special type of mycorrhizal fungal partners that are very interesting in and of themselves. Orchids are actually amazingly successful plants, even though in this area in the Midwest, you don't really see a lot of mycorrhizal fungi. You have to go tropical. And if you go tropical, they're all over the place. But orchids are very, very interesting. We'll talk about that a little bit. And then only 2% are ectomycorrhizal relationships, which I tend to forget that because when I was a PhD student, I worked with ectomycorrhizal fungi, so I thought they were it. But turns out the arbuscular mycorrhizal fungi definitely win in terms of sheer numbers. And then there's another group of fungi called ericoid mycorrhizae that hang out with like heath plants, heather plants. And so ericoid, only about 1.5%. And so the pie chart's nice, kind of trying to put it all into perspective. And then non-mycorrhizal, you see about 8%. The AM, NM, that 7% in the gold. I think they're talking here about some plants form the arbuscular mycorrhizal and then they'll go non-mycorrhizal. So it's kind of like they can shift between having a partner or not. Okay, so here, this is not showing all the different mycorrhizal fungi. It's only looking at three types, the ectomycal, arbuscular, and ericoid. But I just put this up here to show you some traits that you can think about for these different groups of mycorrhizal fungi. I'm not going to get into detail on these. I'm just trying to show you that there's a range of different fungi that are involved. And so you can see that here. So there's different fungi. For those of you who know your fungal classes, you can see that those are all different. And then you can see some of the benefits showing here across the row here. And then also the last row, you can see what kind of plants hang out with these different mycorrhizal types. And so here, all of our trees here, ectomycorrhizal fungi love to hang out with our trees here, but so do a bunch of arbuscular mycorrhizal fungi. In the tropical rainforest, you get a mix, and like one tree can have both. So a tree can hang out with ectomycorrhizal fungi and arbuscular. It can be a mixture. It doesn't have to be. It can be different species of fungi. You know what movie this is from? Okay, so I'm not dating myself yet. You guys still know about Lord of the Rings, right? I watched those movies over and over when my boys were younger. So I thought that was very appropriate. You shall not pass, because it's often shown in readings or sources that by the way, the arbuscular mycorrhizal fungi are also called endomycorrhizal fungi. It's the same thing. So that's why I put the picture on this little column here. But often they'll say the fungi penetrate the root cells, and it drives me nuts. Those of you who've had classes with me before know what that drives me nuts. And so the fungi can certainly be very intimate inside the root, but they don't penetrate into the root cell. If the fungi did that, if they actually got into the root cells where they're like floating around in the cytoplasm, the plant would kick in its defenses and kill it. It's too, the plant's like, hey, respect my boundaries. You get past my cell membrane, I'm gonna kill you. And so, and there's so many readings that put that the wrong way to make it sound like they're in the cells. They're in the tissue. That's different. Okay, so when we say they're in the root tissue, that just means they're in between the cells when you look inside the root. So here's a picture trying to show that a little bit. And this one's just comparing the ectomycorrhizal fungi on the right versus the left is showing arbuscular mycorrhizae. So this is a root, a single root in longitudinal section. And the reason that they're called ectomycorrhizal fungi is all this black stuff that you see growing on the root surface. That's fungal hyphae. Very abundant. You can see it with the naked eye once you train your eye if you look at some roots. And so it's very thick. It's like mushroom. It looks like when you eat mushrooms, that's just fungal hyphae. That's really tightly pressed together into the fruiting body. And so it's kind of that idea. I'll show you some pictures of that. And then you can see that some of the hyphae are going in between the cells. And that's correct. And then those of you in my mycology class this semester, boy, is it a good season for fruiting bodies out there. They're everywhere. I found tons this weekend when I was out hiking. But many of the fruiting bodies that you're seeing with Dr. Robertus's class, a lot of those are ectomycorrhizal fungi that are sending up their fruiting bodies in order to do their sexual reproduction. So lots of those to look for this fall. Okay, and then on the left, they're showing some differences now. With the arbuscular mycorrhizal fungi, they don't form that mantle over the surface, but they have a lot of biomass that goes into the root tissue. So you can see the reason they're called arbuscule mycorrhizal is that they form these branching hyphae in between the cell wall and the cell membranes of the root tissue. And arbuscule, if you look up that word, it means dwarf tree. And so those little branching hyphal sections actually look like a little tiny dwarf tree when you look at it, especially with an electron microscope. But there's no mantle or, fungal tissue on the surface really. They just have some loose hyphae coming off. Same with the ecto. You can see that there is hyphae coming off and growing out into the soil. So depending on which group, you're going to see some distinctions like that. There'll be some differences on how they interact with the root. Here's some ectomycorrhizal fungi, some different viewpoints. So if you dig up a root or if you go to a tree that's been blown over, if you have a hand lens with you in particular, you can go up to the roots and look at them. And so the darker brown, like this is just root tissue here that you see. And then this lighter kind of tannish reddish color, that's the ectomycorrhizal fungi hanging out on the root surface. And so that's actually fungal hyphae so tightly growing together that it is, it does look like the mushroom tissue that you eat on your pizza or something. So that's a nice physical protection because the fungal tissue is so thick that it's really hard for any creepy crawlies to make it through that and get to the root in terms of, attacking the root. So they provide some nice physical protection and they can also provide chemical protection. They can exude certain antibiotics and other chemicals. I really like this lower left image too because it's showing the ectomycorrhizal, the fungal tissue surrounding the root, but then it's also showing very nicely all the hyphae coming off of that. And so again, and then here on the top right, you're seeing some soil that's been very carefully excavated. You can see the mycorrhizal fungal hyphae going out into the soil. This lower right image, so the left image was enlarged. This lower right image is what you'd see with the naked eye. So if you take a section of some of the finer lateral roots from a tree, for example, and clean it off carefully, you can see it's very characteristic that let's see, I don't want that. Let me just see if I can blow that up a little bit. It's very characteristic around trees here when you're looking for ectomycorrhizal fungi. Oh, it's blurry. You'll look at the fine roots like this and you can see they're thicker and covered with that fungal hyphae in that very tissue-like organization. And then they often form a Y pattern shape. It's a little bit easier to see in this in this top left here where it's been enlarged. You can see the Y shape. That's a dead giveaway. You have a ectomycorrhizal root. And so if you see that, because most root hairs and roots, they just grow straight. And so for them to bifurcate like that, it's actually the fungus exuding some chemicals that makes the root do the Y shape like that. So that's a dead giveaway. Really fun to look for. I used to drive my boys crazy because we'd go out hiking and I'd be stopping at every upturned root, tree root, and they'd be like, come on, let's go. But yeah, you can see them with the naked eye once you train your eye. And there's lots of different colors. They're really fun to look at. On the other hand, if you dig up a plant that has endomycorrhizal fungi, you're not going to see a whole lot. In fact, the action of uprooting the plant out of the soil probably takes away any of the hyphae that is on the surface because it's so light, not much there. And so you'd have to go into the lab and look at it under the microscope to prove to yourself that there's endomycorrhizal or arbuscular mycorrhizal fungi there. And these are what you're seeing are roots under the microscope. And so this one here, you can actually see the top right, you can actually see the arbuscle I was telling you about. Looks like a dwarf tree. And then they also form vesicles. It's just all fungal hyphae. So they used to be called VAM fungi, VAM, vesicular arbuscular mycorrhizal fungi, which is a mouthful. So they shortened it to VAM. Okay. Now they're just kind of getting rid of the V, and now the published literature, they usually just call them, I don't know, do they talk to them? AM fungi? That seems weird to say for me. AM fungi? I like VAM fungi. That makes, anyways. So here's again under the microscope, lower left. You can see the arbuscles and then the fat vesicles. Again, it's just all fungal hyphae. And that's a stain that you can do. I've had students in plant phys do that stain. That's just a trypan blue stain. The roots stay clear and the fungus pops blue like that. And then this is under the electron microscope where you can see that dwarf tree, the arbuscle look to it. So you have to go into the lab and look under the microscope to prove to yourself. With ectomycorrhizal fungi, you can see that thick growth on the surface of the root really easily. So those two that are very heavily studied. Orchids, they are really interesting in that the orchids, if you've ever seen orchid seeds, they're like the size of a dust particle. Not kidding. There's just like hardly anything to them. They don't have any carbohydrate reserve in the seed. So if they don't have the correct fungal partner, that seed is toast. It's not going to make it. And it's really kind of bizarre that a plant would adapt that strategy. And yet, orchids are very common. They're a very abundant group of plants. But they just have this very odd situation where um, they have to totally rely on the correct mycorrhizal fungus to be near the seed. If it's the wrong species, it's not going to happen. The seed's done. And so the fungus has to essentially colonize the seed and feed that the germinating seed some carbon. And so the fungus is like totally feeding the orchid seed initially as it germinates and starts to grow. And so the orchid is actually totally relying on that fungus, and the fungus is totally it's a one-sided relationship at the start. And so it has to be the right species. So when you are out hiking around and you see, like, we have a lot of like lady slippers and very beautiful, but when you see one, you should count yourself lucky in this area anyways. They're not super abundant, and they had to have had the exact right mycorrhizal fungus at the exact meet up where the seed is, and all the other environmental conditions had to be correct for that seed to germinate and grow. So it is quite the prospect. It's always amazes me that orchids are so successful, especially in tropical areas, because it just seems like quite the ordeal in order to make it to adulthood. So they have interesting, I showed you that previous picture of how the fungus interacts with the roots. The orchids have very interesting internal systems. So here you see an orchid root, and they're called, well, the fungal hyphae is really tightly coiled. This picture on the right, you can see it a little bit better. And so those are called, oh, do I have it written? Oh, yeah, in the title, orchid pelotons, which makes you think of the bicycle exercise bike. I don't know. It's like, hey, you stole that from mycorrhizal fungi. But anyways, so orchid pelotons, that word pelotons is this coiled formation of the fungus. So it's very distinct. Like the endomycorrhizal don't quite do it like that. The hyphae can grow and die, and so it's a very dynamic fungal life cycle in the root tissue. And the plant is continually getting nutrients from the fungus as it's growing and dying, growing and dying. So very, very interesting. But so once the plant can make it to the point where it can photosynthesize, then it comes a more mutually beneficial relationship. But initially, it's the fungus carrying the responsibility, so to speak, of getting that orchid to the point where it can start doing its own photosynthesis and start feeding back to the fungus. Okay. So I could go on and on and on about mycorrhizal fungi, but let's get into another group. Is there any questions on the mycorrhizal fungi? We cover it in Plant Phys and Plant Kingdom, Applied Plant Science to a little bit more detail. But for this class, I want you to know some basics. And then actually with probably all these groups of organisms that we're going through, we're going to come back to them in context of the different habitats of plants. So this is more like you're getting an intro, and we'll talk a little bit more about them later, including the endophytes. So I've talked about this in my other classes, including microbiology, but so unless you've been living in a cave, you've probably heard about how we are well colonized, right? Humans are well, and other animals, well colonized by microbes. Good ones. We're not talking pathogens here. We're talking the ones that coexist with us from, well, some even say before birth. So as you were an embryo in your mom's uterus, they say you were getting colonized. That's still up for debate. There's some interesting debate about that in the published literature, but and then going through babyhood into adulthood, you're well colonized. And they're starting to get some bit of understanding on that, but they have a long way to go. Plants also have endophytes. We talked a little bit about this already last week. And so basically, we're talking about those microbes that hang out in the plant tissue and they're not the ones that are causing disease. Okay, they're not the plant pathogens. Now, I consider endophytes the ones that stay in the plant tissue for their whole life. So some people say, well, mycorrhizal fungi, they're endophytes. I don't say that because mycorrhizal fungi put their hyphae out into the soil. So they're partly out of the plant and partly in. And so they deserve their own special category. They're mycorrhizal fungi. In most people who study endophytes consider endophytes that are organisms that live their whole life in the plant without causing disease. So that can be beneficial for the microbe because the microbe can rely on the plant host to get its needs met. It gets shelter. I mean, being out in the big bad world is kind of scary for those microbes, I would imagine, so to speak. They have a lot of risk. But if they're in the plant tissue, their risk goes down. They have shelter, they have access to nutrients from the plant body, and the plant can get some amazing benefits depending on what endophyte you're looking at. There's many endophytes that help protect the plant or from pathogens like, pathogenic fungi or bacteria. There's some that will kick out chemicals, alkaloids in particular, that, the fuzzy bunnies and deer don't like, and they'll move on. So herbivores get persuaded to move on with the help of some endophytes. Abiotic stress like drought, heavy metals, super high heat. Again, the presence of the endophytes can be crucial. And the endophytes can help to some extent, especially the root tissue endophytes can help with making sure the plant gets its mineral needs met. And they're still working on all the other stuff. I mean, it's a very, very active area of research, just like all the microbes that hang out with humans. That's a big, hot field of research right now. So it's the same in the plant world. They're trying to figure this all out. They have a lot to go. So these endophytes are within the plant body long term. So they live with the plant, they coexist with the plant through the plant's life. And so it's a stable, kind of long-term relationship, which also surprisingly can get passed into the next generation, at least some of these endophytes. And so there have been some really nice studies that show conclusively that when the seed is getting formed by if you think about angiosperms, where, the flower was pollinated, you get fruit production. As those seeds are getting produced, the microbes from the mother plant can migrate to the seed and become part of the seed. And so they're in there. They're in the seed, in the tissue of the seed. And so then when that seed gets dispersed and germinates and grows, it comes with its own suite of microbes from its mother. And they think that's crucial for that seed to germinate and survive. It's really, an important thing. So this image is trying to show that you can have endophytes getting into the seed tissue. So this has led to some interesting questions on, when you buy seeds, sometimes they'll tell you to surface sterilize the seeds. And although the surface microbes aren't endophytes, and granted, some of those surface seed or surface microbes might be pathogenic, but that's fairly rare. Now it starts to bring up the question, maybe the microbes on the surface of the seed we should also be careful about and not necessarily surface sterilize your seeds before you plant. And then there's, of course, the endophytes in the seed. So personally, I don't surface sterilize most of the time, with the idea that those are probably important. This is one of the published studies showing visually the migration of microbes. So if you are a plant and you are a typical plant, where do you think most of your endophytes are probably coming from? What part of your environment? Where would you guess there'd be a lot of microbes hanging out? Soil, absolutely. The soil is incredibly chock full of microbes. 1 gram can have up to 1 × 10⁹ cells in that gram of soil. And so the soil, they believe, is the biggest source for endophytes. Could they blow in by the wind and get deposited on the leaf or the stem? Absolutely. And they can show that that does happen. But they believe so far anyways that the majority of microbes are coming from the soil. So in this paper, they labeled the bacteria green. So they're popping green. All the red is the plant tissue. And they're showing progression A, B, C, D, E, F, E, F, G, the progression from the lateral root and the root hair. A and B are showing that, and the taproot is C. And then D and G are through the base of the stem. And then E and F are inside the leaf. And so they're showing that the labeled microbe that they put into the soil system could then go endophytic all the way up to the leaf. So I thought that was a very, very nice study because that was a big question. I mean, this was published, I don't have the year on there. When this was published, that was a huge question. Like, can they move once they get into the plant? Are they moving or are they just staying put where they first get entrance? And that kind of has, and they're showing, yeah, a lot of these microbes are moving. And so they think that's a pretty common strategy. So these plants, a typical plant will have all these different endophytes. We're still learning about, who's there and what are they doing. But there's more and more papers studies being published showing in their studies that they're providing all kinds of benefits. So we'll come back to endophytes, especially when we start looking at some of like flower production and nectar production. We'll think about that a little bit in terms of how that may influence pollinators and pollinator behavior. So we'll think a little bit more about this down the road. Okay, so mycorrhizal fungi, endophytes, and then you have just the microbes in the soil hanging out with the plant roots nearby. You have all of that going on. So that leads to this idea, plant growth promoting rhizobacteria. So they are often labeled, or the acronym is PGPR. Lately, though, they've been modifying that to PGPM, where M stands for microbes. So rather than rhizobacteria means root-associated bacteria, but I've seen more papers being published where they're just saying microbes instead of rhizobacteria. So just be aware of that. They mean the same thing pretty much. And so this is like a really big umbrella term, like technically endophytes would fit in this, mycorrhizal fungi fit in this. It's a really big term idea. And so this image is trying to get at all the different things that these PGPRs are doing for the plant. I would argue the fungi should be in here too. That's why I like the micro PGPM. I'm actually favoring that lately because you have to think about the mycorrhizal fungi too. So they're showing several things here. On the left, it's the direct mechanisms, and on the right, it's indirect. So we talked a little bit about what's going on the left, just helping the plant get access to nutrients and water. I'm not going to go into detail on some of that. You don't need to know some of that unless you're really curious. I'll talk to you separately. The indirect, uh, So here, this prevention of, plant disease ISR responses, that is kind of, it's this idea that the microbes that associate with the plant can actually influence the plant's immune system in the sense that we know that microbes can actually tell the plant, so to speak, chemically warn the plant that there's certain pathogens nearby. And that can kick in the plant's, it's called induced systemic response. ISR is induced systemic. And so the microbes are actually, it's pretty much like they're sitting next to the plant in the soil going, hey, we got pathogens here, kick in and start defending yourself, which is pretty amazing. And so these are long-term coexisting microbial partners that are able to trigger the immune response in the plant. So that's what's going on there. Yeah, there's quite a few things in the boxes here that you don't need to know the details of, but definitely showing that you can have many positive impacts on the plant, both directly and indirectly by this group of microbes. And then they're showing also with the red arrow down here that you have them, the PGPRs are also directly killing off or inhibiting the plant pathogens. So these PGPRs are not random in the sense that the plant is actually recruiting certain microbes and favoring certain microbes over others. It used to be thought that plants just kind of sit there and do nothing in the sense of they're totally at the mercy of what's going on around them. That used to be like when I was an undergrad, that was the thought. And now they know that's absolutely not the case. The plants are actively recruiting, in this case, actively recruiting beneficial microbes and actively trying to kill or inhibit the pathogens. And we'll definitely talk more about that in this class. Here's another image trying to get at how these PGPRs are benefiting the plant. And I guess the main reason I put this one in here is this blow up here, where they're showing that the microbes are actually influencing the plant gene expression. And so all the way up to the genetic level, the plants can be affected by these microbes. We'll talk a little bit more about that later in the semester. And that's true for humans and other animals. We know that the microbes that coexist with us affect our gene expression and can affect our behavior as well. So these PGPRs, these microbes that are hanging out with the plant, and then I found this paper published a couple years ago. So arbuscular mycorrhizal fungi enhancing mineralization of organic phosphorus by carrying bacteria along their hyphae. And so the mycorrhizal fungi are growing a little bit on the root. They grow into the root tissue, and then they send out all those hyphae into the soil. In this study, they did both a lab-based and then a more artificial lab-based study, and then a greenhouse, I think it was a greenhouse study using real soil, where they could show under both conditions that with the help of the fungi, the bacteria that could really take phosphorus in the soil and make it super available to the plant, the bacteria would skate along, so to speak, the fungal hyphae and get to these patches that the scientists had set up of phosphorus. They'd have like clumps of phosphorus nutrients. And the bacteria would skate along the surface. The fungal hyphae has a thin film of water, and they swim in the water and get to the phosphorus and solubilize it and give that to the fungus, which then gives it to the plant. And let me see if I can show you. So this is the summary of the paper. So the hyphae going out into the soil, so extraradical just means it's the hyphae going out into the soil, and transport the phosphate solubilizing bacteria to the patches of phosphorus. They move on a thick water film formed around the fungal hyphae. However, the bacteria cannot be transferred to the patches of phosphorus without an energy source from the fungi. So the fungi are actually feeding, probably just, kind of exuding different mineral or different nutrients that the bacteria can continue to take up as they're swimming along to have enough energy to get to the phosphorus and then solubilize it. So it's this amazing partnership. And of course, in the end, the plant benefits. So let me see if I can. So this is an image showing the fungal hyphae. And it's really hard. I'm going to see if I can get the video. I gotta do this. There's no volume. Let me just show this real quick. So this is Pseudomonas putida, very common soil bacterium. And this is on agar, so it's easier to see. Movement is in real time. You see them skating along? So they're on that thin water film on the hyphal surface, and they look like they had a pot of coffee. That's real time. That's not sped up. I was surprised that's not sped up photography or videography. So they're making their way to those phosphorus patches, helping break that down, make it so it's available to the plant. Fungus takes it up, gets it to the plant. I just thought that was awesome. So you have these really interesting interactions going on that, there's so much yet to discover. We don't know the half of it. Whoops, wrong one. So the link, that YouTube link, I didn't post that, but it's in the PowerPoint if you want to see that again. Let's see if I can. Okay. Okay, so these bacteria and these fungi are hanging out in the soil and can be very beneficial to the plant. We're not going to focus on the plant pathogens. That's like a whole nother course. So we're going to focus in on the beneficials. But when you think about the soil environment, the typical plant has its roots in the soil. And if the minerals like phosphorus or nitrogen or potassium or calcium, if any of that is complexed with another molecule and precipitates out of the water solution, typically the roots can't get access to it. So if the minerals are either attached to the soil or they're precipitated out and just kind of a lump in the soil, the roots really can't do much about that. They can't access it. They have to have minerals that are dissolved in solution. And that's what this graphic is trying to show. So on the left, you have kind of a mock-up of some soil particles that have different components to it there. It kind of looks like they're trying to show some clay and other anyways, it's a soil particle. Yeah, clay and organic matter. And all those balls, the different colored balls, are different minerals that the plant needs. And then what they're trying to show is that somehow those have to get off the soil surface and into the water solution of the soil. Once that happens, then the plant has access and can take up those minerals. But if they're on the soil particle, attached to the soil particle, or if they're precipitated out of the soil water solution, the plant has a very hard time, if not, or just impossible time, of getting access to those minerals. So if you've taken soils class or taken plant phys with me, this should look familiar. So this is the idea that when you look at soil systems, the soil particles are negatively charged. So that means positively charged minerals can hang out and attach to the soil. Okay. So at some point, though, the plant needs access to those positively charged minerals because certainly plants need potassium, magnesium, calcium, everything being shown there, they need that. Another thing to factor in, it's being shown on the right. When you look at soil systems, most soil systems are slightly acidic, pH 5.5 to 6.5. And if you look at that range on this graphic, pH 5.5, looks like it's right there to 6.5. You can see that most of those bars of the different minerals are pretty fat in those areas. Manganese is a little not so much, but most of the others are pretty fat in that range. That means they're more available in that range. I'm not going to go into the details of that. If you take plant phys, you've had that of why that's the case. But that's important to realize that under that pH range, most minerals are more soluble. They'll get into the soil water, they'll dissolve, and the plant has a lovely time of getting access to it. But in order to get the minerals dissolved into the water, there's a couple of things that are going on for that to happen. And the first is being shown in that image, which is cation exchange. So if you've had soils, I know Dr. VG or whoever taught it last year, they go through cation exchange process in that class. I cover this in plant phys too. And so this idea that as water is percolating through the soil, there's going to be dissolved minerals in that water. And as it's percolating through the soil, you're going to get some cations that get kind of essentially bumped off or exchanged with another one that can take its place. And that has to do with the binding affinity of these different minerals, which I'm not going to get into. The bigger picture here is showing cation exchange. In this case, the artist drew potassium is percolating through in the water as the water is moving across that soil particle. There's some dissolved potassium. Potassium bumps off, it looks like in this case, calcium. So that's actually cation exchange that they're showing. So once the calcium gets bumped off, gets exchanged off the soil particle, the calcium is now in the water solution, and now the plant can take it up. Does that make sense? Okay. So this process is continually happening as water is moving through the soil system. And that's great for the plant because this exchange process allows for different minerals to get exchanged and into the solution, and then the roots can take it up. So that's one process. But don't forget, the microbes are also really, really important. So yes, cation exchange process, critical. I'm not denying that, but microbes often get not mentioned when they talk about this whole idea of how do the roots get access to minerals. And so microbes, they have their own agenda, they're doing their own physiology, but as they are living their life in the soil, they can often break down minerals. They can break down rocks. Fungi are awesome at that. They can, slowly start to excrete different enzymes and break down the rock material that releases minerals into the solution. And so fungi are awesome at that, but microbes in general in the soil, they are busy solubilizing the minerals for their own needs, but then there'll be some left over for the other organisms, including plants. So you often hear it, they'll say microbes weather the rocks. They're essentially breaking it down. Some people say it's mineral dissolution, same basic idea, rock degradation. So depending on what resource you read, you might hear these terms. And so don't forget about them. They're busy living their life, and in the process of what they do, they can make the minerals accessible in the water, and then the plants can get access to that. Okay, so in some cases, if there wasn't cation exchange and microbes, the plant would have, and depending on the soil system, the plant would have a really hard time flourishing. I've done experiments where you autoclave soil and kill off all the microbes compared to the same soil, its natural microbes. Huge difference in how the plant fares. The plant usually, in fact, most of the time the plant doesn't even make it to flowering stage. It just kind of dies. And so very, very important. So don't forget about the microbes. They're very, very busy. And a lot of their actions benefit the plants. And then this slide is just, again, showing cation exchange. Here they're showing a little bit of why the soil is slightly acidic with, when you look at plants in the system. So we're talking about soils that have plants there, not just barren soil. So plants give off CO2 just like we do. If that CO2 mixes with water, which it would in the soil, you get protons. And remember, pH is dependent on the proton concentration. And so you start, the more protons, the more acidic. And so that's one reason why soil is slightly acidic. And then it can be used to bump for cation exchange. It can be used to bump other minerals off. So just the action of the roots breathing, so to speak, can result in cation exchange. How many people have had soils in this class? Any? No, more than I thought. Awesome. Okay. All right, let's take a break for 10 minutes. So we'll come back at 5 after.
Assuming they will bring on. No. Well, yeah, she'll break it off. Thanks, Donna. I think so, I had a dash.
But sometimes we'll have a no car and
So. Wasn't small. This one came.
Wait.
It should. I felt honestly about the day.
Yeah.
But then you look at like. This one. Which one has the. It's a little. Yeah. So that's. That's a one. One sound, but. I think that's around. Say we play hell, we'll watch what people last and that's right next. That's so far, it's like, yeah, we'll be back. Right, good. So is that this year? That's also bad, but that's me playing or something, but I got a patient. But no, it's not. I think it's long, I think it's distance, and it's massive, on these platforms too. Or a lot of. Well, you don't look great office. Yeah. Part of my issue. No, it's a mom cubs. It's the art increase. Yeah. Bears off in. She's fine.
Yeah.
Yeah. You want to shoot them. Yes.
It's not.
All right, looks like everyone's back. I have a question for you. When I post readings, how do you want me to post them? So I did this, this is the same reading that I asked you to read last week. And I can either give you the URL, the website where right where the article is located, or create a, make it a PDF and upload that. Is there any benefit of one to the other? You like the PDF? Okay, that's usually what I do, but I didn't know if maybe the URL gave some advantage that I'm not aware of. Oh, oh, yeah, I didn't, yeah, especially if you're off campus, right? Good point. Okay, I'll do the PDFs moving forward. All right, let's see if I can switch everything back to the way it was. Okay. All right, so getting back to topics here. So plants rely heavily on their microbes in and on them in the soil. Cation exchange process, really important for the mineral acquisition, but so are the microbes. And so, if you wanted to use this knowledge on your own, whether it be your house plant or if you're growing plants for your job or whatever, you can't ignore the soil. And I'm finding more and more, businesses are becoming very well aware of that, and are actively adding in microbes as inoculants into the soil to improve the plant growth and to improve, like, for example, with CBD production or THC production. Some growers are getting savvy to the idea that the correct mixture of microbes can change the profile of the chemicals the plants are producing, including, commercially ones like the CBD and the THC, where you can make a profit off that. So they also can influence other chemicals too, including helping the plant defend itself, which, when you look at these chemicals that people use plants for, a lot of them are secondary metabolites. And so that's what we're talking about, that the microbes can influence the production of secondary metabolites, both the quantity and quality and ratio. And I'm finding more and more growers are really getting up on that, which is encouraging. It's really nice to see that. And so understanding your soil, what kind of soil do you have? So if you take a soils class, you'll get all into that. And that's super important because, of course, the typical plant is getting so much of its nutrition, its minerals from the soil. And then, the studies are showing getting the right mixtures of microbes into the soil system can really benefit the plant. And so this can be done through amendments. Often I'm seeing products being sold as probiotics for your plant and prebiotics for your plant. And all of these are legitimate. I've seen good published studies. My postdoc professor also did some nice research with prebiotics, which is, in this case, for plants, it would be the idea of like adding in starch into the soil. And starch supports the growth of the beneficial bacteria and fungi, which then help the plant. And so if you've taken a prebiotic, any kind of, they often sell a bunch of like different gummies where they're prebiotics and probiotics that you can take. The prebiotic is something added in to help the microbe grow. It's not directly for you. So in this particular case for the plant, the study that my postdoc advisor did is she put starch into the soil, and the starch fed the good bacteria, which then helped the plant. And she showed a statistical significance. And so I'm starting to see more of that now, that people are getting aware of this, that it's not just using all these synthetic fertilizers, that you can add in the beneficial microbes and the prebiotics to support them. And so I really like this quote. I think it's so true. We don't grow plants, we grow the soil, and the soil grows the plants. And so again, with a typical plant, if you have a good understanding of the soil system and what's happening in there, that's what it takes to grow good plants. And it's really heartening for me to see more and more people are getting aware of that. The old school way of doing like conventional farming or growing, for example, was just hit the field with a bunch of synthetic fertilizers and call it good. And that was the thought for decades. And it kind of works, but long term doesn't. And so when you think about synthetic fertilizers, that they're often put into the soil system at too high of doses, believe it or not, synthetic fertilizers can kill off microbes in the soil, beneficial microbes. It can be very harmful. In fact, they point to some agricultural fields that have been so well or so frequently hit with synthetic fertilizers that they refer to the soil as a microbial desert. It's so low in microbial populations, it's like a desert. And so, yes, it can immediately feed the plant. So when you get these synthetic fertilizers into the soil, the plant can immediately take it up. Yes. But if you don't know what you're doing, overdo it, you can harm the microbes in the soil from that application. Organic fertilizers are the kind of the compare and contrast. So organic fertilizers are not as quick acting because the plant usually doesn't directly take that up. It's usually applied in the sense in the form of amendments like manure or something like that, which has to be broken down first by the microbes, and then that can get fed to the plants. And so it's a slower process. Some growers do both and see better results than if they just rely on synthetic. Some organic farms, of course, have to do the organic, strictly organic. And so it's a little bit of a slower process. But that organic fertilizer does allow for the formation of good populations of the microbes. And that's what you need long-term for the health of the soil and ultimately for growing healthy plants. And so you want to keep that diversity and the populations of your microbes up. And so allowing for that kind of habitat for the plant where they have this nice, healthy mixture of microbes in their soil next to the roots allows for really good growth by the plant. And you also have to think if you are ever into growing your own garden plants, for example, I know that's becoming more and more popular, which is great. Have you ever heard of companion planting? Okay. Back when I introduced this term like a decade ago, or maybe a little longer than that, that was like, what? What are you talking about? It's good to see some head nods. And so companion planting is this idea, and it's still, there's a lot of research that needs to go on with this, but it's the idea that you plant groups of plants together in one soil system and that the particular plants you pick, they actually support each other really well and all the way to, flower and fruit production, if you're thinking about angiosperms. And so often one of the partners that you'll pick is a plant that hangs out with nitrogen fixing microbes. So I wanted to talk a little bit about this idea and what I mean by that. Probably the most famous example and the one that stretches back probably the longest time frame is the Three Sisters planting Native Americans have done for centuries, where traditionally it's corn, squash, and beans that are planted together. And so the corn plant grows very tall, of course, which provides a nice physical system for the beans because the beans are a vining plant. So the beans will vine around the corn stalk. So it helps the beans stay upright and access sunlight. And then the squash has those big, lovely leaves. The squash grows more horizontal and can cover the soil. Those leaves cover the soil, create a nice, cool, moist, humid situation that helps suffocate weeds that are trying to grow there. And then they're showing that the bean plant is providing nitrogen into the soil, but it's really not the plant itself per se. It's the microbes that hang out with the bean because the bean plants hangs out with nitrogen fixing bacteria. And so this is a classic example of the companion planting idea. And I really, I'm hopeful to see that there'll be more of this kind of research going on. I know organic farmers are catching on to this idea. I've spoken to a couple in the Midwest area where this has been their strategy for their some of their fields in their farming operation, and it's been working out really well. So if you have bacteria hanging out that can produce nitrogen, enough nitrogen to help the plants, that's great because then you don't have to spend money on fertilizer. You don't have to worry about what over fertilizing with nitrogen can do to your soil and to the water table below it. So let me just talk a little bit. I'm not going to go into the same level of detail that I cover in Plant Phys, but I do want you to be aware of this idea. This is a very special group of plant growth promoting rhizobacteria. It's the nitrogen fixers. And so you're looking at plant roots right now. And these are, I remember the first time I saw these types of roots, I thought the poor plant was infected and felt really bad for it. And then the teacher said, no, this is a good thing. And so those are root nodules that are forming. That's plant tissue. It forms into this spherical shape, mostly spherical. And I think you can kind of see in that image where I circled that the kind of looks reddish brown, and that's accurate. It should look that way. At least some of them you can see that color. But those are nodules, and if you cut that open and look at it, there's tons of bacteria in there hanging out. And so they have a nice cozy environment in that plant tissue, and they're busy doing nitrogen fixation. The picture on the right is just a different plant species, and you can see the nodules look a little bit different, but same basic idea. But those look like tumors of some sort. And it's easy to think that it might be a bad thing, but it's a good thing. So those are root nodules. And so that's the area where the plants, where the bacteria are hanging out with the plant. And just last year, a published paper, 2024. Well, I posted the summary of the paper, not the actual paper, so it's a little easier to read. But please read that posting. It's in the readings and videos. So up until today, actually, I always taught that this is a prokaryotic only thing. Nitrogen fixation can only happen with a prokaryotic cell type, which means bacteria or cyanobacteria. Those are bacteria too. But just bacteria. Bacteria can do nitrogen fixation. Other organisms cannot. And today is the first time I get to say we were wrong. So there's a recent discovery of a eukaryotic cell. It's an algae that has what they've termed a nitroplast organelle, which was once a free-living cyanobacteria. And so this is a huge deal. Like I wouldn't be, well, it probably wouldn't get a Nobel Prize because it's having to do with plants. They often are very dismissive about plant research. But anyways, that's a whole nother thing. So anyways, the nitroplast, they're terming it the nitroplast. And so it is, and that little posting that it's a small reading that summarizes the paper on this, and it goes into how they can call it an organelle. It's officially now an organelle. So they had to do a lot of research on this. I guess this was discovered like 25 years ago, but they had to do like a lot of research to verify this, to really truly call it an organelle. So this is the first time where you can say a eukaryotic cell can do nitrogen fixation. That's a huge deal. I can't believe I'm actually saying that. Yeah, I just didn't think that that was going to happen. So please read that. It's pretty straightforward. It's not the actual paper. It's just a summary of it. So they kept it nice, easy to read, no jargon. So mostly prokaryotic, but just be aware that there are this case. So it's an algae. So that's an algal cell. And I don't know if you can see that black arrowhead. You see that from where you're sitting? Okay, so it's pointing to this big thing in there. That's the nitroplast. So that was once a free-living cyanobacteria. So this is the endosymbiosis idea of that was free-living, but it got incorporated into this particular algal species and over time has become the nitroplast through evolution and adaptation. So, but we're going to focus mostly on the idea that you have bacteria in the soil and bacteria hanging out in some plant roots that can do nitrogen fixation. Not all plants have this ability to partner with these bacteria. Growers would love it if some of the major crops that we grow, in fact, they're trying, we'll talk a bit about that toward the end of the semester, how they're trying to make plants partner with nitrogen fixing bacteria, and they are getting some success with that, which is amazing. But anyways, the reason these bacteria are so special, they're very special, is that they have this enzyme, dinitrogenase. And what that does is it takes the triple bond between two nitrogen. So dinitrogen, we have 80% of it in our atmosphere. Plants can't touch it directly, but these bacteria have this particular enzyme that breaks that triple bond and creates a reaction that ends up with ammonia, NH3. That ammonia quickly gets incorporated typically into amino acids for the plant. So hugely beneficial for the plant to have this partnership with these bacteria. You can have bacteria hanging out in the soil that just do it on their own. They're just living their life and they're fixing nitrogen. You can have bacteria that hang out in the plant tissue, in those nodules that are doing this nitrogen fixation and giving that ammonia to the plant. So that's the main one that we're most interested in for this class, is the ones that form the relationship with the plant. So this is just an example of different plant species. On the top there, you can see the top portion of the table, and you can see there's different bacteria listed. You don't have to know this to any detail. I'm just showing you that there's different plants out there that can do this, including some important crops, sugarcane, obviously, pretty important. But there's, plants that are aquatic that also have that azolla. And then on the bottom, these are just some bacteria that just do it on their own, and they can just hang out and do it. Okay. But the ones that hang out with plants, they actually, and I'm not, in Plant Phys, I go through this in detail, but basically what I want you to know is that there's a lot of communication between the plant and the bacteria, where they realize they can both benefit from this relationship, and the plant will allow the bacteria to, they actually use the word invade. It's kind of a bummer they chose that word because it sounds negative. It's actually a good thing though. And so the bacteria will invade the root hair of the roots. And so all the bacteria have been labeled green here, and the plant tissue is red. And so you can see they're outside kind of on the tip of the root hair, and then they make it in and they start progressing down the root hair until they can get to the bulk of the root tissue. So here they're free living, they're attaching to the root hair, and then they start to grow and move down the root hair to get to the root tissue. And so this is all done through a very complicated communication back and forth. The plant needs to know that these bacteria aren't pathogens. And so that it has to be established first, and then they'll allow for this invasion. And so here's a mock-up on top trying to show that process. The red cells there are the bacteria progressing down the root hair. And the goal is to get to the more bulky part of the root, and then the plant starts to build those nodules, and the bacteria hang out in there, and they're busy doing nitrogen fixation. So the picture on the left is just an outside visual of a root nodule. And then if you stain it and look at it under the microscope after you slice it in cross section, all the green are different bacteria in the nodule. And so the plant allows, creates a house, essentially a home for these bacteria, and the plant can get access to the ammonia that the bacteria make. So the bacteria, pull dinitrogen from the air, and you don't have to memorize this reaction. The main point is that you have to expend ATP to get this to happen. So this is not something that the plant does for free or the bacteria does for free. It's costly, but it's worth it because once the plant forms this relationship, it's a win-win. The bacteria benefit from it and the plant benefits from it. But it's not free. And so the way growers often determine how much does this cost the plant is they'll look at how much carbon is required in order for this to happen. And again, you don't need to memorize this, but 12 grams of carbon needed to fix 1 gram of dinitrogen. That's a fair bit of carbon. So it's costly. The point that this slide is making is it's costly. It costs the bacteria 16 ATP to run this reaction, and the plant has to feed the bacteria in order to get this to happen. So they're both investing in it, but the payoff is worth it. So nitrogen fixation, we'll talk a little bit more about this. As I said, when we get toward the end of the semester, we'll look at the future of agriculture and forestry and the idea of what they're trying to do with the microbes and nitrogen fixation. Any questions on nitrogen fixation? I know those of you who've had Plant Phys have got it in way more depth than this, but you don't need to know it to that depth in this class. Okay. So this slide set, we went through a lot, but basically the overarching theme is plants and their microbes, their immediate surroundings, what's immediately in the soil around them, what's in them, what's on them. And I wanted to kind of talk about this early on in the semester because I want you to keep that in mind as we're working through the rest of the content. Because you can't ignore these other players. Just like for your own health and your own well-being, you can't ignore the microbes that hang out with you. They're realizing that now too. So wanted to bring up one other point before we shift totally away from plants and microbes. So, is that the rain sounding like that? Is that like hail? Does that sound loud? Okay. Huh? Is it just the wind? That's pouring rain though too. Oh joy. Just think if you have that for mycology tomorrow. Is it mycology tomorrow or boreal flora? Which one is which? Boreal flora is Friday. Oh, fisheries management. Do you go outside for that?
Put the ball
Ooh,
for
get to go to the bog. Hopefully no one will fall in this time. I would just love to shadow Dr. Robertus's class. Like I've been meaning to just step into his mycology class and go out with all the all the students, and I just can never get it to work in my schedule. Such a bummer. Um, okay. I haven't put this up into Educat because I didn't think we'd get to it yet. But there's a couple of things I wanted to say about this. I will upload these slides. Oh, let me do this. Okay, how many people have taken evolution? Okay, most people. Do you remember? Might be a very hazy memory, but do you remember hearing anything about the hologenome theory of evolution? Does that, yes, no, kind of, a little bit? I don't know how much they talk about it who teaches it now in our department? Okay, I'll have to talk to her, see how. Um, because I did want to bring this up and come on. I don't know why it's doing this. Okay. Yeah, I'll have more to say on that quote through this semester. Plants get short shrift in the grand scheme of Earth, even though they dominate the Earth. All right, hologenome concept. So, the host and their symbionts. So, whether you're talking about yourself and all the microbes that coexist with you through your life, or a plant and its microbes, that is the holobiont. And in the hologenome, typically, if you really want to be picky, that word, the hologenome, is the host genome plus the microbiome genome. So you're zooming into the DNA. What does each partner have to offer in terms of the DNA? But sometimes I tend to use that word kind of hologenome. I tend to think, I kind of have it take the place of holobiont. It's just, I don't know, easier for me to say, I guess. But anyway, so you have working together, the host and all the microbes. They function as a unique biological entity and therefore also as a level of selection in evolution. And so this is a team of scientists that have been presenting this theory or hypothesis for quite some time now. And it's slowly getting into some of the other areas of evolution, but it seems like it's not widely talked about. Um, So the Rosenbergs, they came up with, they said, okay, in order for this to be accurate, to act at the level of selection and evolution, they are saying that four things have to be met for this to be true. So now I'm going to go through those four. All animals and plants harbor abundant and diverse microbes. We know that to be true. That's been proven. And the second sentence is also very true. And at least when it comes to humans and plants, that the number of microbes and their genetic information far exceeds that of the host. You don't have to write this down because I'll post it. The second thing they say has to be true. It has to be transmitted from one generation to the next with fidelity. And we were just talking earlier how seeds contain their own endophytes and surface microbes on the seed coat. So that is met. And when you were born, in fact, when your mom went into labor, you were already getting colonized right during the labor process. And some say in the womb, you were already getting colonized. So it's true for humans too and other animals. Number three, the microbial symbionts and the host interact in ways that affects the physiology, health, and fitness of the host and the microbes within its environment. And lots of studies are showing this to be true. For sure, with humans and other animals, as well as with plants. The fourth concept, genetic variation in the hologenome can be brought about by changes in either the host genome or the microbe genomes. Under environmental change or environmental stress, the microbiome can adjust more rapidly and by more processes than the host, and so can enhance the evolution of the holobiome. This is true for us too. Microbes that coexist with us actually can adapt quicker to changes, whether you change your diet. That's a big environmental change for you and your microbes. So just something as simple as that, changing your diet, the microbes in you can adapt much quicker and help you and benefit from that, to some extent, at least, depending on what your diet is. And so we'll talk about these four concepts and give some examples in the plant world mainly. So it's not the strongest of the species that survive, nor the most intelligent, but the ones most responsive to change is a famous quote by Charles Darwin. So he's saying, I have something in a plaque in my office that says adapt or die, and that's pretty much it. Yeah, if you can't adapt, you're out of here. You're going to go extinct, right? So the point here is that the microbes that coexist with you, whether you're a plant or a human, can help you do that. Does this sound at all like, does this get covered in evolution? Not, maybe not so much. Well, I'll talk to a little bit. Okay. Okay, I think I'm going to stop there. The next topics are a little deeper and take too much time. So I'm going to take this slide set. I think I want to tweak it just a little, and then I'll post it either later tonight or tomorrow morning. And I'll probably maybe give one reading on this, but I got to find a good reading, a more up-to-date reading, that's not too long and too jargony. Okay, so I will be posting something. Let me find that. If I can find something newer, it's a little easier, but we'll stop there for today.